Hello there. Here to talk to you about our next topic. It's a little bit of a rainy day outside here, but I'm under the carport, and it's nice and pleasant temperature-wise, so I thought it'd be nice to be out here instead of stuck indoors. So what I want to talk to you about is a little bit of cell biology, and we're going to start by talking about diffusion, which is an important process in a lot of biological phenomena. And so we need to talk about the cell membrane and then movement across that membrane, and then we'll talk about diffusion, and then in the next lecture we'll talk about osmosis. And so every cell is surrounded by a membrane. That's what defines the outside of the cell. And so here's a figure from your book. And this is true for both an animal cell and a plant cell. So now plant cells have a cell wall on the outside of that membrane, but they still have the membrane that defines the outer edge of the cell that contains the cytoplasm. And animal cells have the membrane but do not have a cell wall. And so, by this idea of a cell membrane and what it's made of, it seems like we kind of make a big deal about that in biology, but it is a big deal because that defines the inside of the cell from the outside of the cell. Well, duh. Why is that a big deal? It's because all cells must regulate their internal environment. And so all living things live in an environment that's constantly changing, and some environments change more than others, but relative to the inside of the organism and the inside of the cell, the external environment is always changing, yet the inside of the cell needs to stay fairly constant. And so all living things need to be able to control what gets into their cells and also what gets out of their cells. If you can't control what gets out of your cells, then your cells are just kind of leaking stuff all the time. That's no good. And you can't just let anything into your cell. So it's a very important concept, and it's important to an idea called homeostasis. Homeo meaning same, stasis meaning static or same too, I guess. Homeostasis is maintaining a constant internal environment. And why do we make such a big deal about this? Because this is one of the characteristics of life. And so there are a lot of parameters that must be maintained internally, and homeostasis is a huge topic we could talk about for a long time. Here's a figure from your book that doesn't, just shows a bird sitting in a tree during a cold period. And just to give you an example of maintaining a constant internal environment. So for this bird, the body temperature inside the cells is the same, even though it's cold outside. But if it were warm outside, their body temperature would stay the same. Well, that's true for homeotherms. Not all organisms maintain the same body temperature, but there are lots of other things that need to be kept constant within the cells, like the water levels, how much water concentration, salt levels. So when we say salt levels, we're not talking just regular table salt, but we're talking all kinds of dissolved ions, all kinds of molecules that are dissolved in the cytoplasm in the cell. For proper life processes, you've got to have a certain constant level of those things, and that does not happen spontaneously. Living things must expend energy to maintain this internal environment. And so we talked about this already, about sunlight energy being captured by photosynthetic organisms, and then that energy gets passed to all the other organisms. Well, this is a huge reason you need energy. This is what you spend tons of energy on, is just maintaining this internal environment. Again, when you lose the ability to maintain homeostasis, then that's when you slip away and die. So maintaining this internal environment is a big deal to living things, and consequently, controlling what crosses the cell membrane, that's an important idea to maintaining that constant internal environment. So consequently, crossing those membranes is a very important idea in biology. So that's why we're talking about it today. Now, something we haven't yet talked about, but is interesting and important, is what does it mean to be a living thing? What is life? This is a biology class. We're studying living things. But as with any discipline, you need to define the basics, like what exactly is a living thing. And at first blush, you could say, well, it's just kind of a funny question. I know, what's alive and what's not alive. I know that a rock is not alive and things like that. But it's important to clearly define the subject you're studying. That's what makes it interesting. But also, as we'll see, they're organisms. It's not always clear. Are they alive or are they not alive? And so there are several definitions or characteristics of living things, and maybe you see a little bit subtle differences depending upon the class or the textbook. But we're going to go with what your textbook has, and there are several characteristics of living things. And so here's the whole list right here. So we just mentioned homeostasis. That's a very important one. And so living things must maintain homeostasis. They must maintain an internal cellular environment that's different from their external environment. Living things share common types of molecules. So we talked in an earlier lecture about biological molecules, carbohydrates and lipids and proteins and nucleic acids. And all living things use those different kinds of molecules. So it's common to all of life. All living things are made of cells. Lots and lots of organisms are just one single cell, right? Bacteria are just one single cell. But all living things, that's the basic building block. So again, that's why we're talking about cells today. And that's also why no matter what you want to study in biology, you need to understand what's going on at the cellular level a little bit. Growth. All living things are bound by birth and death, and they grow. And so they start smaller than their adult form, and they grow into an adult form. And maybe they don't grow very much, but all living things do grow at some point. Movement. So this movement can be a tricky one, and I think we could argue about it a little bit, because obviously animals move, but you say, what about plants and that? But plants can move to follow the sun, and so that's movement. So we won't belabor that one too much, but that's probably true of all living things. Reproduction and passing on of genetic material. This is a big one, right? So all living things come from other living things and also then reproduce. But when you reproduce, you pass on genetic information. And so, that's interesting. That's something that's going to be unique to living things. All living things respond to the external environment. And so they keep their internal environment different from the external environment. And so they also change if the external environment changes. And so a lot of that sometimes it just means you've got to do something different to maintain, your internal environment separate. But responding to an external stimulus the external environment is important to living things. Another biggie that sets living things off is metabolism. And so this is what we talked about earlier. Harvesting energy, breaking apart those bonds, taking the energy that's released when you break those bonds and capturing that energy and making new molecules, or capturing that energy and using it in chemical reactions to do different things. And then also disposing of wastes. And so all those complicated reactions that make a living thing that a living thing must do to stay alive, that's all considered metabolism, and it all uses energy. And finally, populations of living things can evolve. Now, when we talk about evolution, we're going to stress this very much. Individuals don't evolve. So an individual organism does not evolve over its lifetime, but populations of organisms do evolve. And so what that means is that on average, the population can change in characteristics over time, but individuals don't evolve. And so that's a lot of things, but you need to have all these things in order to call something alive. And so, like I said, this can become important when you talk about organisms where it's not clear whether or not they're alive. And a good example of this are viruses. And so this is a great time to talk about viruses, right? Because we're in the middle of the COVID pandemic, and COVID is caused by a virus. And so we need to talk about, well, what is a virus, and are viruses alive? And so do we consider viruses living things? And so this has been something that, as throughout, once viruses were discovered, and then we figured out that viruses cause many common diseases, then the question is are they alive? Lots of things that make you sick, that cause disease, are living organisms, right? And so if you get a bacterial infection, bacteria are living things. And so if you get a bacterial infection, that can make you sick, right? You can get a fungal infection. Fungi are living things. They can make you sick. You can get a parasitic protozoan infection, like malaria is a organism that lives in your red blood cells. And so those are diseases caused by other living things. But it's never clear, is a virus considered a living thing? But first let's talk about what is a virus, so we can understand how these things work. Basically, viruses are very, very simple. They're just some sort of genetic material, either DNA or RNA, so some sort of nucleic acid wrapped in a protein coat. And it's usually not a huge amount of DNA. It's only a handful of genes. We said humans have 25,000 genes, and viruses usually don't have that many genes at all. And that's it. That's there's important proteins, also inside that protein coat, but basically it's just a protein ball with some genetic material. And so this is a figure from your book showing a magnified image of the SARS virus. And so the SARS virus is related to the COVID virus. They're both called coronaviruses. And they're called coronaviruses because if you look at the image here in the microscope, you see lots of little spikes sticking out. They call them the protein spikes on the surface, and it makes it look like a crown. Corona means crown. And so this is a group of viruses that looks like a crown because of these spike proteins. And SARS, you may or may not remember, but a few years ago there was an outbreak of SARS, and it was pretty much over in China, and it didn't get to the pandemic level like we're getting with COVID, but it was a similar virus to COVID. And then before that, it was MERS, some Middle Eastern respiratory syndrome, was also a coronavirus. And so these spikes that make the crown, they're important to how this virus functions. Now, not all viruses have these kind of spikes. Viruses have proteins on their surface, but not all of them look like this. So this is kind of what makes the coronavirus family a little unique. And so, like I mentioned, other coronaviruses, MERS, and of course COVID is a coronavirus. Those spike proteins in COVID are very important because they're used to bind to cells in your respiratory tract. And so you know that coronavirus is a respiratory disease, and that's how it affects you, and that's where it's located in your body. And it's these spikes that allow it to bind to your cells. Again, that's not unique to this virus. All viruses do this. All viruses need to bind to the membrane, the cell membrane of their host. And when they bind to the cell membrane of their host, then they can get into their host cells. And when they get into their host cells, they release their genetic material into the host cell. So here's this virus, which is a protein coat with some genetic material into it. It sticks to the cell of a host, which allows it to dump its genetic material into the host cell. That's how all viruses work. So again, we're talking about crossing the membrane, getting stuff into the cell. And the viruses have figured out a way to sneak into the host cell. And once they dump their genetic material into the cell, the virus hijacks the cell. It takes over the cell's mechanisms. And it uses the cell of its host to make copies of itself. And so again, the virus is just some genetic material, it's either DNA or RNA, and a protein coat. Well, all cells have genetic material, all cells make protein, and so all cells have the capability of making copies of this virus. And so that's what the virus does. It's figured out a way to sneak into the host cell and then use the host's mechanisms to make thousands and tens of thousands of copies of itself. And so it copies all the DNA thousands of times, and it makes a whole bunch of protein coats, and then it repackages all those new copies of the genetic material inside all these new protein coats. And so now the host cell is just filled with thousands and tens of thousands of copies of this virus, and eventually the cell bursts and releases all those copies. Of course, that kills the cell, and then now you've got a whole bunch more copies, and they can go and infect other cells in your body. And each of those cells can make more copies, and so on and so on. But also, all those copies of viruses can leave your body when you cough, when you sneeze, when, whatever, and then get picked up by someone else, and now the virus does the same thing. It gets into their cells and makes copies of itself. So that's how all viruses work. And coronavirus is no different, but coronavirus is, some viruses are better at getting into your cell than others. The coronavirus is really good at getting into your cells. And so that's one of the reasons that makes it deadly. And there's lots of other reasons that make this a very bad virus to catch. So anyway, this is figure 1.4 from your book. Study this again so you can understand the process here. But that's basically the big idea, that viruses are able to sneak through the cell membrane, and then they take over the host cell. And so it's all about crossing that membrane. Obviously, obviously the host does not want that virus to come in. But this is an example of where the host is unable to keep things. Again, living things are all about controlling what gets into their cells and what leaves their cells. But this is an example of something that is able to circumvent that, and so that's why they make you sick. That's why it's a problem. Okay, so the question is, are these viruses alive? And they have some characteristics of living things. And so you see that, like they use common types of molecules, right? They use DNA and RNA and protein, just like other living things. Movement, they're able to move, right? They're able to move within an organism because the cell bursts and the viruses are able to spread to other cells, and then they're able to move from one host to another host, so they can move. They're able to reproduce and pass on genetic material, right? That's what they're doing inside your cell. They're getting inside your cell and then making copies of themselves. That's reproduction. And that reproduction includes nucleic acids. It includes this genetic material. I've circled response to external environment in yellow. I'm not sure about that. Maybe they do, maybe they don't. I mean, I think it's possible that under certain circumstances, the viruses might change their protein coat and wait for more favorable outcomes. But I'm not a virologist, so I'm not sure about that one. That's why it's circled in yellow. So maybe they can respond to the external environment, maybe they can't. I don't know about that. And then the populations can evolve. And again, we'll talk about this later, but that's an important factor when you talk about viruses and pandemics, is that viruses can, change. Populations of viruses can change over time. And that's sort of what happened with COVID, is you had these different viruses, but then different viruses can get packaged together and create new viruses and then the viruses are better at getting into your cells and things like that. So these are all characteristics of living things. And so you say, okay, well, viruses have all those characteristics, but they're lacking others that are important to living things, and those are the ones circled in red here. So viruses cannot maintain homeostasis. They don't really control what gets into and out of their cell membrane. I don't know if they really have a cell membrane. They're not made of cells. So yeah, I guess I answered my own question. They don't have a cell membrane because they're not made of cells. And so when we talk about the structure of the cell, you'll see that the cell has a typical membrane, and there are certain things inside the cell. Viruses lack those. They don't really grow. They don't start off, a virus doesn't start off small and then grow to an adult virus. It's just the virus just uses the host cells to make copies of itself as is. So they don't really have growth. And the biggie, probably the most important thing that tells us that they're not really living things is that they don't metabolize. They don't harvest energy and use that energy. The virus cannot reproduce without its host, and it uses its host's energy and its host's mechanisms. And so that is kind of a good one to watch out for, as far as if you're trying to decide if something is alive. Does it break apart molecules, use the energy that's released to build other molecules? Viruses do not do that directly. They have to use their host. And so for these reasons, viruses are not considered to be alive. But you can see why the question is interesting. Because, until you break it down, until you clearly define life, you can see that there's some things about viruses that make you wonder, is this a living thing or is it not a living thing? To me, they're just interesting. They're kind of in that twilight zone. They're they have some characteristics that are kind of living, but they're not really alive. But at any rate, they're interesting. Okay. And so back to internal regulation. One of those characteristics of living things is that they can control what gets into their cells and out of their cells. And they try to maintain this constant internal environment. And so that's the topic that we want to talk about the rest of this lecture. And so we're talking about crossing the cell membrane. We need to kind of talk about the structure. What is this membrane made of? First off, we want to point out that cell membranes are called semi-permeable, right? Which means that some things can cross the membrane, but other things cannot cross the membrane. And so permeable means, it can cross, anything can cross. But semi-permeable means only some things can cross. Well, again, that makes sense, right? If we want an internal environment, we want to let some things in and some things out, but not everything in and everything out. This keeps the inside of the cell different from the outside of the cell. And we've already mentioned the important structure of cell membranes, and those are phospholipids. And so you remember a phospholipid is a fat, and fats usually have 3 hydrocarbon tails, but in the phospholipid, one of those tails has been replaced by a phosphate. So that's phosphorus with 4 oxygens. And you remember that that lipids as a class, they are hydrophobic. They fear water, right? And so fats and cholesterol and things, they don't mix well with water. That's a general characteristic of that group of molecules. But in the phospholipid, you'll recall that only one end of the molecule is hydrophobic, and the other end, where that phosphate is hydrophilic. And so that's really important for building a cell membrane. And so hydrophilic means it loves water. And so if you've got a bunch of these things and you mix them in water, what are they going to do? So let's say I've got 10 million phospholipids and I dump them in water. What's going to happen? Half of those molecules, one half of each of those molecules is going to love the water. The other half is going to want to get away from the water. How are they going to arrange themselves? Well, it's pretty interesting. They'll form a bilayer. And so when we talk about the cell membrane, we say it's made up of a phospholipid bilayer. So bi, two, layer. So two layers of phospholipids. And so if you look at this diagram here, you can see that there's two layers. And what happens is that the hydrophilic heads point in either direction because they're fine with water and they're sticking themselves into the water, but the hydrophobic tails can tuck to the middle part of this bilayer. And so you see you got one layer where the hydrophilic heads are pointing to water, and then the other layer is flipped. And again, the hydrophilic heads are interacting with water, but in this way, those hydrophobic tails can tuck to the center part of this, sandwich. And so those hydrophobic tails can hide from the water. And that is the basic structure of cell membrane. And so just think of like a soap bubble or something. It's just going to be a sphere, but if you zoomed in on it at the molecular level, this is what it looks like. And so there you go. And so this is the semi-permeable membrane that surrounds all cells. Now there's a lot more to this membrane. And as we go along, we'll talk about there's lots of proteins that are stuck in this membrane and lots of proteins on the surface of the membrane. But the basic structure, the scaffolding, the foundation of this is this phospholipid bilayer. And so, one side of the membrane is outside the cell and one side is inside the cell. And so then this is how the living thing tries to control what comes across this membrane. And so let's talk about movement across membranes in just a little bit. So what needs to get into a cell of a living organism? Like oxygen, water, food particles, right? We need to get that energy in, stuff like that. You got to get that into the cell so that you can work on, so that you can do those chemical reactions that make you alive. What needs to get out of a cell? Well, whatever's left over from those chemical reactions. So carbon dioxide, you got to get rid of. Sometimes you got to get rid of water. If you've got too much water in the cell, you've got to get rid of it. Waste particles, again, what's left over from those chemical reactions, you need to get rid of. And so those are the kind of things that come in and out of the cells. Think about what you do every day. What does it mean to you? What you're alive, what do you do all day, every day, right? What do you take in? You take in water every day. You take in food every day. You take in oxygen every breath. What do you get rid of every day? You get rid of water when you urinate. You get rid of waste when you urinate. You get rid of carbon dioxide every time you breathe out, right? That's stuff that you're doing all the time. But why are you doing that? It's because in your cells, you need that stuff or you need to get rid of that stuff. So consequently, those are the kind of things that need to get across that cell membrane. And so how this happens, that's again a big deal in biology. These are not the only things, but you get the idea. And of course, there's other things that want to get into your cells, like viruses, but maybe you don't want them in your cells. So again, keeping bad stuff out is something that living things do. But as we see with viruses, they're not always successful. Understanding how things cross a membrane is a big deal in biology, and it helps to explain all kinds of stuff. And so that's why we're talking about that idea right now. Well, an important way to get things into and out of cells is a process called diffusion. This is not the only way, but this is a very important way. So that's why we're talking about diffusion in this lecture. So let's do that. Okay, so we got to just define some terms here so that we're all using the same language. So stuff that gets dissolved is called the solute, and stuff that does the dissolving is called the solvent. And in here, most of the time the solvent is water, right? So any liquid could be a solvent, but in biology it's water. And so things get dissolved in water. So solutes get dissolved in solvents. Okay, so if I put some solute into a solvent and I don't stir it, I just put it in there and walk away, what's going to happen? So like you're dissolving Kool-Aid in a glass or something. You just put it in there and add the water, but you don't stir it. What's going to happen? Is that Kool-Aid just going to stay there in a lump? Or if you put your sugar in your coffee and you don't stir it, is the sugar just going to stay there? No, it doesn't stay there. It's going to spread around. Now, if you stir it, it spreads around faster. But if you just put it in there and leave it alone, eventually that solute will spread throughout the entire container, right? And so let me show you just a visual representation of this. And so this is just a little program, and I'll run little models here and there it's a program called NetLogo, and it just allows us to do little to model things and kind of see what happens. And so I'm going to show you one of these NetLogo models right now to illustrate what I'm talking about. Okay, so here's just a little model to kind of illustrate what we're talking about. And so this is just supposed to simulate a container here, and each of these dots is a molecule. And the molecules in this model behave like molecules in real life. They each have kinetic energy. They move around at random. They bounce off of each other. They bounce off the sides. And so it's mimicking what molecules do in real life. And so when I click Setup, so I've concentrated them. So you see that they're all concentrated. There's a high concentration here and a low concentration around the outside. And so watch what happens when I hit go. And it's what you expect. You can see that they're all moving at random. So maybe just pick one and kind of follow it. And you see they just kind of bounce off of each other. But the net effect is that they started concentrated and now it's spread out evenly. That's diffusion. I'm going to do it one more time. So now there's a high concentration in the middle, but there's no extra energy added here, and there are no other properties other than just random movement. Okay, so what you saw there is just a very basic visualization of what happens at the molecular level. So molecules move around at random. Molecules are always moving, right? They always have some kinetic energy, which means they're always moving around and bouncing off other things. But there's no direction to it. They just move at random and bounce off of things at random, right? Because all molecules move at random, they tend to spread out evenly. And the tendency of molecules to spread out evenly, that's called diffusion. And so things diffuse out. You've kind of heard this term. You can picture this term. And so again, if you just put some Kool-Aid or some food color in a drop in a glass of water, without stirring, you're going to see that color or that Kool-Aid is going to spread throughout the whole container. That's because every molecule of that food coloring or every molecule of that Kool-Aid is moving at random, which causes them all to spread out evenly, as we saw in the model. And so, at first, the molecules of the solute are concentrated at first, but if they all move at random, they all spread out evenly. That's diffusion. Now, an important idea behind this is it does not require a direct input of energy. So remember I said, you put it in the cup and you don't stir. You don't add any extra energy. It will still spread out. If you add energy, it spreads out faster, but you don't have to. And so there's no direct input of energy. These molecules have all the energy they need, and they will just spread out randomly over time. Okay, so remember that we're talking about this in the context of stuff crossing membranes. So what happens if we add a membrane to this concept? So for example, the molecules are concentrated on one side of the membrane. What's going to happen? Well, first let's ask what happens if you have an impermeable membrane. You remember that cell membranes are semi-permeable. I'm saying let's talk about an impermeable membrane. So a membrane that molecules cannot cross, but you've got the solute concentrated on one side of this membrane. What's going to happen? So let's just take a quick look at that situation in another model. So here we have a similar model to the one I showed you earlier. And again, you've got these molecules here that will just move around at random. But this time, we've got a membrane separating the container. And all the molecules are concentrated on one side of the membrane. So we have a high concentration on the left side of the membrane and a low concentration on the right side. In this particular model, if you look here, you see that this is an impermeable membrane. So this molecule cannot cross that membrane. And so what happens when they start to move? You see that they spread out evenly on one side, but not the other side because they can't cross the membrane. And so here again is what we just saw. You've got an impermeable membrane, and on one side you've got molecules concentrated. And so without any extra energy, those molecules will just move at random and they will spread out, but they'll only spread out on one side of the membrane. They can't cross that membrane. When they hit the impermeable membrane, they bounce off. And so you've got molecules that are spread out evenly on one side and still nothing on the other side. Okay, same idea, but now let's put in a cell membrane, a semi-permeable membrane. And so now the solute we're looking at can cross the membrane. Remember, semi-permeable means not everything can cross, but in this example, the solute we're working with can cross that membrane. So everything's exactly the same, except now the membrane is semi-permeable. So let's take a look at that. Okay, in this last example, we've got the same setup as last. We've got the molecules concentrated on the left side of the membrane. This time, however, it's a semi-permeable membrane. So these molecules can cross this membrane. And so we have a high concentration on the left side and a low concentration on the right side. So we have a concentration gradient from high to low as you go from left to right. So now, again, the molecules are just going to move at random. And let's see what happens. And so, as you expect, they spread out evenly as if that membrane was not there. But of course, the net effect is that you had a net movement of molecules from left to right. At the start, all the molecules were on the left and none were on the right. And so now you've got
about an equal number on both sides. So there was a net movement from left to right. And if we put them back in motion again, you can see they don't stop moving, but you tend to have as many move in one direction as in the other. So there's no net movement at this point, but there still is movement. And if we run it again, see now they're concentrated on one side and nothing on the other side. And so again, the molecules are only moving at random. There's no other forces in effect here. And as they move at random, if I stop it here, you see there's still a higher concentration. There's more on the left than on the right. But there's some on the right now. And as they move at random, they start to spread out. And at this point, you have about an equal number on both sides, an equal concentration on both sides. And so the molecules moved down their concentration gradient, and they spread out evenly, and that's diffusion.
And so here again, we've got a semi-permeable membrane. The solutes concentrated on one side. They'll spread out, but this time instead of bouncing off the membrane, they just go right through it. And so it's as if that membrane was not there. And so the molecule spreads out evenly on both sides. The molecules move at random, and they're spread out. They diffuse to both sides of the membrane. And so again, that's diffusion. But it's the sort of diffusion that we see associated with cell membranes. And so what happens is you've got molecules that were concentrated on one side of a cell membrane, and they moved to the other side. And you didn't have to add any extra energy, just random molecular movement meant that they would naturally just spread to the other side of the membrane. That's diffusion. And we say that the molecules moved down their concentration gradient. And so what does that mean? Well, when we started, you notice on the left side of the membrane, you've got a high concentration of the solute. And on the right side, you've got a low concentration of the solute, right? And so we say that this is a concentration gradient. A gradient is a slope, or anything that changes over distance. And so here you've got high concentration that changes to low concentration over distance. That's a concentration gradient. All those molecules that are concentrated will just move at random, and they will spread, and they'll end up spreading out to where there was a low concentration. That's moving down your concentration gradient when you move from high concentration to low concentration. And so again, well, why do we care? So we're just talking about molecules moving at random. Big deal. Well, this is a very important way that stuff gets into and out of your cells. And so, for example, when you breathe in, you take in a ton of oxygen. You have a high concentration of oxygen outside your cells, but you need it inside your cells. How's it going to get there? Well, the oxygen's just going to move at random, and it's going to diffuse from the high concentration to the low concentration. And so it's going to diffuse from outside your cell to inside your cell due to this random molecular motion. You've got a high concentration of carbon dioxide inside your cell. You got to get rid of that. How are you going to get it out of your cell? It's just going to diffuse out by random molecular movement. You've got a high concentration inside your cell, a low concentration outside your cell. The molecules are going to move from high to low, and they're going to exit your cell. And so lots of stuff crosses your cell membrane by just diffusing. Again, this doesn't require any extra energy. It happens because of that random molecular movement. But it always happens from high to low concentration. And so things will always spread out from the high concentration and diffuse to the low concentration. Okay, now once those molecules spread out and have diffused down their concentration gradient, do they stop moving? Yes, no, no way to tell. Well, molecules never stop moving, right? Until you're at absolute zero, there's always some movement. And so the molecules keep moving, but they move in both directions. Again, just picture these molecules moving at random. And so you've got molecules moving from left to right, just because of their random motion. Some are going to go from left to right, but once they've spread out evenly, an equal number will move from right to left. And so although the molecules are moving, you don't have any net movement. And so eventually the number moving from one side to the other equals the number moving from the other to the first side, but they all keep moving. When you start out, you do have a net movement. And so if you have a high concentration of oxygen outside your cell, it's going to diffuse into your cell. So there's a net movement into your cell. But once the concentration's equal on, inside and outside your cell, the oxygen molecules keep moving, but just as many move in as move out, so there's no net movement. And so there's an unequal concentration, so there will be a net movement from left to right. But after a while, they're equal concentration, so there's no net movement. They're still moving in both directions, but it's equal movement in both directions. That's called a dynamic equilibrium. It's an equilibrium. The concentration is no longer changing, but it's dynamic. Dynamic means moving, and it's because they're moving, but they're moving equally in opposite directions. And so here's a figure from your book, again, just sort of trying to underscore what I've been saying, that you've got this phospholipid bilayer. So here's a cell membrane, and you've got a high concentration of both oxygen and carbon dioxide on top, on the top of this picture, and a low concentration at the bottom of this picture. So both of these will spread and move from the top to the bottom, and you'll have a net movement. And so that's an important way that things can cross cell membranes. Okay, and so it's a fairly simple idea that profoundly affects lots of things in biology. We're going to talk about the diffusion of water, which is osmosis, in the next lecture. And then as we go through the semester, you'll keep hearing this term, and you'll see what we're talking about and why this is important the rest of the semester. So anyway, that's diffusion. So please let me know if you've got any questions, and I will talk to you later.